Basics of Medical Biochemistry. First edition, 2023. Chapter 19. Molecular Genetics, Part 1 of 2. By Professor Dr. Muhammad Jawad. MBBS, Pfau, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College, Air University-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 19. Molecular Genetics. 19.1 Introduction. Molecular genetics is the study of the heritability of traits at a cellular level. It is more concerned with the studies on DNA and the cellular mechanisms of trait transmission between cells. In contrast, molecular biology is the study of living things at the level of the molecules which control them and make them. It is mainly concerned with studies on nucleic acids and the proteins which are constructed using the genetic instructions encoded in DNA. 19.2 Basic terminology in molecular genetics. 1. Autosomes and sex chromosomes. An autosome is any of the numbered chromosomes, as opposed to the sex chromosomes. There are 23 pairs of chromosomes in human cells, for a total of 46 chromosomes. Humans have 22 pairs of autosomes and 1 pair of sex chromosomes, which are X and Y. Sex chromosomes help in determining the sex or gender of an individual. Autosomes are present in almost all of the cells of the human body. The 22 pairs of autosomes are referred to by number basically in inverse correlation with their size. As chromosome 1 has approximately 2,800 genes, chromosome 22 has approximately 750 genes. Figure 19.1 Autosomes and sex chromosomes. Females have 2 X chromosomes in their cells, while males have both X and Y chromosomes in their cells. Egg cells all contain an X chromosome, while sperm cells contain an X or Y chromosome. This arrangement means that the male is responsible to determines the sex of the offspring when fertilization occurs. The X chromosome is larger and carries more genetic information than the Y traits controlled only by genes found on the X chromosome, example hemophilia, red-green color blindness, are said to be sex-linked. Sex-linked traits occur far more frequently in males than in females, since a male inheriting an allele for a recessive trait on the X chromosome lacks a corresponding allele on the Y chromosome that might counteract its effects. Several disorders are associated with an abnormal number of sex chromosomes, including Turner syndrome and Klinefelter syndrome. 2. Complementary DNA, cDNA, and cDNA library. Complementary DNA, cDNA, is synthesized in the laboratory from messenger RNA. cDNA is not genomic DNA, because the transcript of genomic RNA has been processed, i.e. it lacks promoters and introns. The enzyme reverse transcriptase is used to synthesize double-stranded DNA that is a complementary copy of the mRNA. The addition of linker sequences to the end of this DNA, which contains the restriction site, followed by treatment with a restriction enzyme, produces a cDNA preparation with cohesive ends ready for insertion into a vector. cDNA is often used to clone eukaryotic genes in prokaryotes. Preparation of cDNA represents the genes that were actively being expressed in a cell, an organ, or a whole organism at the time of harvesting and is called a cDNA library. Figure 19.2 Formation of complementary DNA from mRNA. 3. Recombinant DNA, rDNA. Recombinant DNA, rDNA, molecules are DNA molecules formed by laboratory methods of genetic recombination, such as molecular cloning to bring together genetic material from multiple sources. It results in the creation of sequences that would not otherwise be found in the genome. Figure 19.3 Recombinant DNA, rDNA. Recombinant DNA technology uses enzymes to cut and paste together DNA sequences of interest. The recombined DNA sequences can be placed into vehicles called vectors that transport the DNA into a suitable host cell where it can be copied or expressed. 4. Exon, intron, and codon. Three common technical terms in molecular genetics, exon, intron, and codon, have specific technical definitions. Exons and introns are features of DNA, whereas codons are features of RNA. By definition, exons and introns are sequences in a protein-coding gene region of a double-stranded DNA molecule that is expressed as proteins, or intervening sequences not so expressed. The exons and introns are typically shown as the single-stranded sequences of the sense strand of the DNA, written 5 rightward arrow 3, left to right. Simply, the coding part of a gene is known as an exon, whereas the non-coding region is termed an intron. Both introns and exons can undergo transcription, but mRNA formed by introns is completely removed by a set of splicing enzymes. The RNA sequences equivalent to DNA exons and introns can be referred to as exon transcripts and intron transcripts, or equivalents, respectively. Genes that contain introns are known as interrupted genes. Codon is a sequence of 3 successive nitrogenous bases in the molecule of mRNA. It is involved in protein synthesis by incorporating a particular amino acid in a protein molecule. It means that each amino acid is represented by 3 consecutive nucleotides sequence called a codon. There are 4 nitrogenous bases in mRNA, and they can form as many as 64 types of genetic codons when they are arranged in a triplet, 43, sometimes presented as a translation table. As there are only 20 standard amino acids to take part in protein synthesis and quite a big number of codons makes it is clear that most amino acids have more than one codon. This feature of codons is called the degeneracy or redundancy of the codon. Figure 19.4 Role of exons and introns in protein synthesis. 5. Penetrance and expressivity. Penetrance refers to the probability of a gene or trait being expressed. In some cases, despite the presence of a dominant allele, a phenotype may not be present. One example of this is polydactyly in humans, i.e., extra fingers and or toes. A dominant allele produces polydactyly in humans, but not all humans with the allele display the extra digits. Complete penetrance means the gene or genes for a trait are expressed in all the populations that have the genes. Incomplete or reduced penetrance means the genetic trait is expressed fractionally in some part of the population. The penetrance of expression may also change in different age groups of a population. Reduced penetrance probably results from a combination of genetic, environmental, and lifestyle factors, many of which are unknown. This phenomenon can make it challenging for genetics professionals to interpret a person's family medical history and predict the risk of passing a genetic condition to future generations. Expressivity refers to variation in phenotypic expression when an allele is penetrant. Considering the example of polydactyly again, an extra digit may occur on one or more appendages. The digit can be full size or just a stub. Hence, this allele has reduced penetrance as well as variable expressivity. Variable expressivity refers to the range of signs and symptoms that can occur in different people with the same genetic condition. 6. Trait. A trait is a specific characteristic of an organism. The genetic contribution to a trait is called the genotype. The outward expression of the genotype is called the phenotype, example hair color or blood type. Traits are determined by genes, and they are also determined by the interaction with the environment with genes. In fact, genes are the messages in DNA that define individual characteristics. So the trait is the manifestation of the product of a gene that is coded for by the DNA. The word phenotype is sometimes used interchangeably with the word trait, and phenotype may also define a whole compilation of traits together. 7. Gene. The gene is the basic physical unit of inheritance. Genes are passed from parents to offspring and contain the information needed to specify traits. Genes are arranged, one after another on chromosomes. A chromosome contains a single, long DNA molecule, only a portion of which corresponds to a single gene. About 30% of DNA is composed of genes, but only 1.5% is involved in coding, while the remaining 28.5% acts as non-coding. Humans have approximately 30,000 to 35,000 genes arranged on their chromosomes. Figure 19.5 Relationship between chromosome, DNA, and gene. A gene could be as short as a few hundred base pairs or as long as many thousands. The BRCA1 and BRCA2 genes, for instance, are very long and huge. The beta-globin gene, on the other hand, is only a few hundred of these nucleotides. A gene is a packet of information coding generally for a protein. 8 allele. An allele is an alternative form of a gene, one member of a pair, that is located at a specific position on a specific chromosome. An allele is one of two or more versions of a gene. An individual inherits two alleles for each gene, one from each parent. If the two alleles are the same, the individual is homozygous for that gene. If the alleles are different, the individual is heterozygous. 9 dominant and recessive gene. Dominant gene refers to the relationship between two versions of a gene. Individuals receive two versions of each gene, known as alleles, from each parent. If the alleles of a gene are different, one allele will be expressed, it is termed the dominant gene. The effect of the other allele is masked and it is called a recessive gene. In Huntington's disease, there is a dominant mutation where, one copy of the Huntington's disease gene is mutated, which is dominant. The individual has a disease regardless of the nature of the person's other Huntington's disease gene allele, which may be perfectly normal. Figure 19.6 Pattern of dominant and recessive genes in Huntington's disease. 10 Genotype. The total gene content of an individual is called a genotype. The term also can refer to the two alleles inherited for a particular gene. The genotype is expressed when the information encoded in the gene's DNA is used to make protein and RNA molecules. This is the internally coded, inheritable information carried by all living organisms. This stored information is used as a blueprint or set of instructions for building and maintaining a living individual. These instructions are found within almost all cells, they are written in a coded language, the genetic code. They are copied at the time of cell division or reproduction and are passed from one generation to the next. These instructions are intimately involved with all aspects of the life of a cell or an organism. They control everything from the formation of protein macromolecules to the regulation of metabolism and synthesis. 11 Phenotype. Pheno simply means observe and derives from the same root as the word phenomenon. This is the outward, physical manifestation of the organism. A phenotype is an individual's observable traits, such as height, eye color, and blood type. The genetic contribution to the phenotype is called the genotype. Some traits are largely determined by the genotype, while other traits are largely determined by environmental factors, such as what one eats, how much one exercises, how much one smokes, etc. 11 Heterozygous. There are two possible forms of a gene for a diploid organism. Each gene is made up of two representative alleles, one inherited from the maternal source, mother, and the other inherited from the paternal source, father. Heterozygous refers to having inherited different forms of a particular gene from each parent. A heterozygous genotype stands in contrast to a homozygous genotype, where an individual inherits identical forms of a particular gene from each parent. The word heterozygous simply means that the biological mother and father, when they contributed their copies of a particular gene to offspring, did so in a way that the DNA sequence is slightly different. It can be different at one point in the gene, or it can be different at several points in the gene. 12 Homozygous. Homozygous is a genetic condition where an individual inherits the same alleles for a particular gene from both parents. Homozygous describes the genetic state where an individual has inherited the same DNA sequence for a particular gene from both their biological mother and father. It's often used in the context of disease, where an individual has inherited a mutant allele or an error in DNA sequence from their mother and they have inherited the identical mutant allele from their father. In this situation, that individual is homozygous for that mutation. They have two identical copies of the deleterious version of that gene and, as a result, they are then going to be predisposed to the respective genetic condition. Figure 19.7 Homozygous and heterozygous arrangement of a gene. 13 Codominance. Codominance is a relationship between two versions of a gene. Individuals receive one version of a gene, i.e. an allele, from each parent. If the alleles are different, the dominant allele usually will be expressed, while the effect of the other allele, called recessive, is masked. In codominance, however, neither allele is recessive nor dominant, rather the phenotypes of both alleles are expressed. In codominance, neither allele can mask the expression of the other allele. An example of codominance in humans is the ABO blood group, where alleles A and alleles B are both expressed. So if an individual inherits allele A from their mother and allele B from their father, they have blood type AB. 14. Genome. The genome is the entire set of genetic instructions found in a cell. In humans, the genome consists of 23 pairs of chromosomes, found in the nucleus, as well as a small chromosome found in the mitochondria. Each set of 23 chromosomes contains approximately 3.1 billion bases of DNA sequence. 15. Gene expression. Gene expression is the process by which the information encoded in a gene is used to direct the assembly of a protein molecule. To do this, the cell interprets the genetic code, and for each group of 3 letters, it adds one of the 20 different amino acids that are the basic units needed to build proteins. Figure 19.8 Gene expression. 16. DNA fingerprinting. DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. There are various methods for analyzing DNA to establish if 2 samples are the same or different. For example, 2 cloned pieces of DNA can be studied in the laboratory to determine if they have portions in common, and thus overlap with one another. In a different setting, such as a crime scene, DNA samples can be collected and analyzed to determine if they match DNA samples obtained from suspects of that crime. If 2 DNA samples have the same fingerprint, then there is a very high statistical likelihood that they came from the same person. DNA fingerprinting is also used to establish paternity. Figure 19.9 DNA fingerprinting, suspect 2 matched with crime scene. One common laboratory method used to detect specific DNA molecules from among many other DNA molecules is Southern blotting, after the name of scientist J. Murray Southern, who formulated this technique. The principle of Southern blotting is based on the process of separation of DNA fragments by gel electrophoresis followed by the recognition by labeled probe hybridization. The DNA fragments are detached based on their charge and size during electrophoresis. Other bold techniques are also available, such as Northern blot used to detect RNA and Western blot to detect protein. 17. DNA sequencing. DNA sequencing is a laboratory technique used to determine the exact sequence of bases, A, C, G, and T, in a DNA molecule. The DNA base sequence carries the information a cell needs to assemble protein and RNA molecules. DNA sequence information is important to scientists investigating the functions of genes. The technology of DNA sequencing was made faster and less expensive as a part of the Human Genome Project. And recent developments have profoundly increased the efficiency of DNA sequencing quite more. Figure 19.10 DNA sequence data from an automated sequencing machine. DNA is a linear string of nucleotides or bases identified by the first letters of their chemical names as A, T, G and C. The process of deducing the order of nucleotides in DNA is called DNA sequencing. Since the DNA sequence confers information that the cell uses to make RNA molecules and proteins, establishing the sequence of DNA is key for understanding how genomes work. 18. Cloning. Cloning is the process of making identical copies of an organism, cell, or DNA sequence. Molecular cloning is a process by which scientists amplify a desired DNA sequence. The target sequence is isolated, inserted into another DNA molecule, known as a vector, and introduced into a suitable host cell. Then, each time the host cell divides, it replicates the foreign DNA sequence along with its DNA. Cloning also can refer to asexual reproduction. Cloning is a molecular process of making millions or billions of copies of a single molecule. It's different from the uses of the terms cellular cloning or organism cloning that are used in the field of reproductive genetics. 19. Epigenetics. The term epigenetics was introduced in 1942 by embryologist Conrad Waddington, who, relating it to the 17th century concept of epigenesis, defined it as the complex developmental processes between the genotype and phenotype. The word epigenetics is of Greek origin and means over and above, AP, the genome. Epigenetics is an emerging field of science that studies heritable changes caused by the activation and deactivation of genes without any change in the underlying DNA sequence of the organism. Epigenetics involve genetic control by factors other than an individual's DNA sequence. Epigenetic changes can switch genes on or off and determine which proteins are transcribed. It is involved in many normal cellular processes. Unlike genetic changes, epigenetic changes are reversible and do not change the DNA sequence, but they can change how the body reads a DNA sequence. Epigenetic changes alter the physical structure of DNA. One example of an epigenetic change is DNA methylation, i.e. the addition of a methyl group, or a chemical cap, to part of the DNA molecule, which prevents certain genes from being expressed. Another example is histone modification. Several lifestyle factors have been identified that might modify epigenetic patterns, such as diet, obesity, physical activity, tobacco smoking, alcohol consumption, environmental pollutants, psychological stress, and working night shifts. Epigenetic changes are responsible for human diseases, including Fragile X syndrome, Angelman's syndrome, Prader-Willi syndrome, and various cancers. 20. Epigenome. The epigenome is the collection of all of the epigenetic marks on the DNA in a single cell. The epigenome consists of chemical compounds that modify, or mark, the genome in a way that tells it what to do, where to do it, and when to do it. Different cells have different epigenetic marks. These epigenetic marks, which are not part of the DNA itself, can be passed on from cell to cell as cells divide, and also from one generation to the next. 21. Biotechnology. It is defined as the method by which a living organism or its parts are used to change or to incorporate a particular character into another living organism. It involves the application of scientific principles to the processing of materials by biological agents. End of Part 1 of 2, of Chapter 19. Thank you.